All right, okay, well, let's get started. Welcome everybody to today's colloquium. It's nice to see everybody in person and also those who are remote. And it's a real pleasure today to introduce Dr. John Poole, and I'm honored to introduce him today in this special seminar. John is, as you know, a member of our faculty. He just a little bit of background, so we learn a little bit more about John's path. He got a bachelor's in science and biology in Western Michigan University. Later, he got a PhD in genetics and evolution at Cornell University in the laboratory of Charles Acuadro, where he started studying the molecular variation and adaptation sweeps in Drosophila, using Drosophila as a model, but on site. So Drosophila, even in Afrotropical and other areas. And that's where he also started working on the idea that, or the idea that Drosophila and developing the idea that Drosophila had migrated out of Africa in the last century. He later became a postdoc in the laboratory of Rasmus Nielsen at UC Berkeley and also University of Copenhagen, and also in the laboratory of Charles Langley shortly at the University of California, Davis. And there he continued working on innovative methods to analyze population inference from genomic variation. He did not only work on Drosophila, he also worked on other models, including mice, where he worked on divergence of species in a hybrid zone, and also in humans and high altitude humans in Nepal. When he joined as assistant professor at UW in 2011, has continued this work, and he became an associate professor here in 2017. He has continued working on the analysis of genetic variation, focusing on population structure and biogeography to understand the adaptation and evolution of key traits. Key traits using, in a sense, a few selected traits like pigment, body size, cold, and wing, cold tolerance, and wing size. He also carried out the high throughput sequencing of 1,000 Drosophila genomes, which has provided a great impetus to the analysis of genomic variation using this model system. So his studies have really led to new methods and insights on the role of genetic variation and its dynamics in during adaptation. And these are really widely applicable to a lot of fields, from biomedical science, because a lot of these traits are going to be polygenic and complex, and to rapid evolution in a changing climate. I also want to thank John for all his service, a tremendous service as a great, wonderful citizen in the department. In the last 5 years, as an example, he has been the chair of admissions. He really has made a tremendous effort and a difference in that role. So thank you, John. Without further ado, I introduce John, and thanks a lot for the seminar.
Thank you, Francisco, for a very kind introduction, and I really appreciate the chance to be back here and to update you all on some things that have been going on in my lab in the last few years. And so I would say probably more than anything, the key to understanding what my lab is about is that we use genetics and genetic variation in particular to learn about evolution. But our primary orientation is to address big picture questions in terms of how does evolution work at the genetic level, and not necessarily the precise details of how it's acted in any particular case. And so the research that I'm going to tell you about today is really aiming at that. It's aiming to get a better understanding about how processes like adaptive evolution operate at the genetic level, but more, like at the level of genetic architecture rather than, in terms of like exactly which precise genetic changes are responsible for which exact trait. So that's kind of, the overall set of questions that we're looking at. Okay. So I'm going to structure this talk into three major parts. The first part is partly about the early history of Drosophila melanogaster and what we've learned about it in just the last couple of years. And then it's about how we can use that model of population history to inform our studies of natural selection. And we'll talk about what we've learned about adaptive trait evolution by comparing populations of Drosophila melanogaster. In the second part of the talk, I'll continue that kind of local adaptation theme. And I'll tell you about three separate instances of the adaptation of Drosophila melanogaster to colder environments around the world. And what we've learned about this parallel evolution, both at the level of genetic variation and also at the level of transcriptomic variation. And at the very end, I'll tell you about some studies that we've recently started to leverage time samples, either from fly strains alive in the lab today or little fruit flies in a museum drawer, and to leverage these temporal samples to get even potentially clearer inferences about how natural selection has acted in the genome of this model species. All right, so I mentioned I'm going to start out with what we've learned about the origins of Drosophila melanogaster. Most of you probably know this species, as a laboratory model, but of course, its original habitat was not a little glass or plastic vial in a lab. This is a real species, and it actually came from somewhere, but we didn't always know where. We know that it's a human commensal species. You probably all realize that. This is often the very fly that might be hovering around your overripe bananas on a warm summer's day. But in terms of the history of the species, at least, when I began my career, things were still pretty hazy on that front. And yet, having a good model of population history is essential for any serious studies of population genetics. And so this was a major failing in Drosophila population genetics specifically. About the only thing that people had figured out was that it was very likely to have originated from Africa. All of the closely related species were either from the African mainland or from nearby islands. But it was a little hard to figure out where Drosophila melanogaster might have come from within Africa, in part because nobody had ever found it in a convincingly wild environment. So early on, there were ideas it may have come from Western or Central Africa. And then by the time I came here, based on simply patterns of genomic diversity, I argued that it was likely to have originated from Southern Central Africa specifically, especially this belt of seasonally dry woodlands called the Miombo and Mopane forests. But that was an idea, and it seemed like these areas had not been had searched as thoroughly as some other parts of Africa in terms of trying to find the species in the wilderness. And so people, would ask me, "Are you going to go and look?" And I would say, "Well, I'm trying to get tenure right now." So I didn't really have time, and I had a lot of other things going on. But lucky for me, my collaborator, Marcus Stensmeyer, was motivated to get out there and give it a try and look for Drosophila melanogaster from these very environments. And he had a slightly different motivation than me. He's not a population geneticist. He's actually more of a neurobiologist, and he studies the olfactory system of Drosophila and of mosquitoes. But basically what he wanted to know is, why is the fly olfactory system the way it is? Why do they prefer the smells that they prefer? What was their original food source in their wild environment? And so that was his major motivation to go and look for these flies in their wild environment and also to figure out what they were eating. And so he did. He went there, and in fact, yes, he found Drosophila melanogaster in the wilderness of Zambia and Zimbabwe in these Miombo and Mopane forests. And not only that, he discovered what is, at least seasonally, their favorite fruit. They are overwhelmingly concentrated in these marula groves during the 4 or 5 months of the year when ripe fruit is available. And so, that's all cool. But what's really impressive is that in one study, he bridged, and his postdoc at the time, Dr. Suzanne Mansourian, they bridged those natural history observations to precise molecular details in terms of figuring out which specific compound in marula the flies are attracted to the most, and which specific fly olfactory receptor is primarily responsible for that attraction. And I only had a very small role in this study, looking at the population genetics of olfactory receptors, but I feel like this kind of sets the stage for what we did as well. It also for why this scenario might have gone into the history of Drosophila melanogaster becoming a human commensal. I mentioned marula as a fruit that the flies are especially attracted to more than any other fruit that's ever been tested. An interesting thing about marula is that this was a very favored fruit resource of the San people, who are the original modern human inhabitants of this part of Africa before agriculturalists and pastoralists expanded their ranges. There were the San hunter-gatherers, and marula was an extremely important food resource to them. And archaeologists have found caves with millions of marula stones built up over time. The San would store the nuts and they would ferment the fruit. And so this represented a very attractive resource to fruit flies and actually aligns with some aspects of Drosophila melanogaster's own biology in terms of their willingness to enter dark spaces and their tolerance of alcohol. It's entirely plausible that marula may have been sort of a gateway drug in terms of Drosophila melanogaster becoming a human commensal species. And we'll kind of come back to that in terms of the estimates of population history that my lab obtained. So that's the part of our own study that I'm going to focus on here. Marcus sent us some preserved flies that he collected from Zambia, from wilderness areas, and we sequenced their genomes. And we compared these genomes to those from town populations from across Africa and beyond. This, by the way, down in the corner is some of Marcus's own art. It's like where the wildflies are, basically. So that was perfect for me because I had a kid who was the perfect age for reading that book too. Anyway, I want to emphasize that this study that I'm about to talk about is especially the work of Quentin Sprengelmeyer, who is a recent PhD student in my lab since graduated. So what Quentin did is he used a software program called FastSimCol, which uses the allele frequency spectrum across multiple populations to learn about parameters of population history. Question.
Yeah. How was true wilderness classified?
So what we were looking for here was basically an area of Miombo and Mopane forest that was miles from any trace of human settlement. So anywhere from 5 to 50 kilometers are the places that we've collected these flies from. Should I change the lighting at all? Is it a little bit? Is it washing out? It's fine. Okay. All right. So this is a method that allows us to estimate important parameters of population history from the allele frequencies that are observed jointly across multiple population samples. And so before I show you the results here, I'm just going to stop for a minute because you might be wondering, like, okay, you've been touching on Drosophila melanogaster history for your whole career. Why would you have waited until 2020 to estimate parameters of their population history? And actually, a big part of the reason that I waited that long is that I wasn't sure it would work. And so, these methods of what we call demographic inference all assume that there is no selection going on of any kind, or basically that the genetic variation that you're analyzing is unaffected by natural selection, either at those sites themselves or at linked sites. And it wasn't clear, how robust the Drosophila genome would be to that assumption. Drosophila melanogaster is a very populous species. It has a very high population size. We know that natural selection is very effective in such species, and it's been argued that natural selection has fairly pervasive effects across the Drosophila melanogaster genome. So that was the concern, is would selection basically bias our estimates of demographic parameters? And indeed, there was one study that specifically argued just that. So they studied a specific model of recurrent genetic hitchhiking involving rare but strong selective sweeps. And they studied how that biased the inferences of historical parameters within a single population across relatively long population genetic timescales. And they suggested that there were, that the situation was pretty bad, that there are strong biases there. So this was motivated by a specific model of recurrent hitchhiking that had been suggested for Drosophila melanogaster. But we also took a look at this question, and we found that actually another published model of recurrent hitchhiking involving many more selective sweeps, but with lower selection coefficients, so weaker selection, but more common selection, that fit aspects of the divergence data better, the divergence between species, that is to say. And that model, this many weak sweeps model, led to much less bias in demographic inference, especially for the parameters that relate relatively recently diverged populations, such as the ones that we're actually studying in this case. And so this study, led by Jeremy Lange, another recent PhD student in the lab, is what kind of gave us confidence to go ahead and do this demographic inference for Drosophila melanogaster, including the newly discovered wilderness population. So that's what I'm going to show you results from now. And I'm going to really just cut to the chase. This is the model that we came up with, basically, where on the left here, we are showing you both the time scale of population split events that we estimated with the x-axis labeled in thousands of years. And on the, on the y-axis, basically, we are showing you the size of the populations for each of these relevant branches, where a wider bar here means a larger population. And so the chronology that we suggest here, which is kind of our number one interest here, is yes, I mean, it does look like, this wilderness Kafue population is kind of like the basal lineage here. And so that's interesting, but we're also interested in the details of how this and when this species has expanded within Africa and beyond, because that's essential for a lot of our other projects in the lab regarding locally adaptive differences between human commensal populations. So in terms of, the timing of this species expansion within Africa and beyond, we have one sample in Egypt and one in France up here. So first, of course, there's an expansion within Africa. So, I'm kind of color coding the more ancient populations from the ancestral range in red here. And so roughly 10,000 years ago is when, we kind of give birth to the yellow populations on this map. And so this entails an expansion through Central Africa and beyond, even crossing the Sahara not long after. It's worth noting that this expansion initiated roughly 10,000 years ago, because this is also, archaeologically speaking, this was found to be the peak of the San people's use of the marula fruit. And so this could make sense, if the flies using this fruit that was being, collected by humans in large quantities, if that was sort of, a joint behavior that enabled the expansion of this species within Africa initially. And so, yeah, I mean, even crossing the Sahara relatively quickly, by roughly 10,000 years ago. And so that may seem like a stretch by the standards of today's climate, but it is worth noting that at that time, the Sahara was not quite as imposing as it is today. And it was more scrub and less desert, I would say, at least. There may have even been some early sort of proto-agricultural behavior among humans in this part of the world. For example, there's evidence of fig cultivation in the Middle East going back 11,000 years. And so that may have made for kind of a soft landing spot for this species in the Middle East. And then relatively more recently, we have the species expanding into some cooler environments. This is a high altitude South African population here. I note that, this is about the time that the Bantu people brought agriculture to Southern Africa. And around that same time, we have the founding of a high altitude Ethiopian population. This EF is my favorite fly population because it has a lot of unique traits, and it's from more than 3,000 meters above sea level, whereas EA is a low altitude population that's actually from a scorchingly hot place. And then, in roughly the same time frame, we have a population split between our Egypt and France samples, which could correspond to, roughly speaking, the expansion of the species from the Middle East into Europe. And that was also a time of increasing fruit cultivation in Europe. So a lot of these events actually seem to line up surprisingly well with known aspects of human history. So the key in terms of the rest of this talk is that now we have an actual model of population history, which helps us to understand what should genetic variation look like, in the absence of any other processes like natural selection. So that's a key element for learning about natural selection, and that we actually use this model in various of the projects that I'm going to tell you about next. And one of the things that we do, downstream of that is we use the diversity of these populations to try to learn about how adaptive evolution works at the genetic level. And so we think that the geographic diversity of Drosophila melanogaster populations, all of which share a recent common ancestor, relatively speaking, gives us some excellent opportunities to study how adaptive evolution works at the genetic level. And so some of the questions we're interested in on this front are things like, when a trait evolves adaptively in a natural population, how many genes are involved? It could be 1, it could be 10, it could be 100, it could be 1,000 or more. And also whether natural selection is primarily acting upon new mutations or else standing genetic variation that was already there. And lastly, given that natural selection is favoring certain genetic variants, are they rising all the way to 100% frequency, or are they stopping before that point? Either because selective pressures changed or because the trait got where it was going and didn't need to fix all of the causative variants. So these are some of the kind of genetic architecture questions that are really our primary focus in studying the genetic basis of adaptive evolution. I mentioned these populations before, and the fact that this species has rather recently expanded into a wide array of worldwide environments. And that gives us a great opportunity to use local adaptation to study the genetic basis of adaptive evolutionary change. Because these are populations that have recently adapted to new environments, but they also share recent common ancestors with other populations. Take this high altitude Ethiopian population, for example. It only went up there, we think, about 1,000 or 2,000 years ago. And so genetically speaking, it's still pretty closely related to this low altitude Ethiopian population, and yet it lives in a vastly different environment. So what that means is that most of the genome is quite similar between these populations, but at genes that have been affected by natural selection, they may depart from that pattern in detectable ways. So it gives us a great system to learn about how adaptive evolution works in general. We have kind of a three-part system for studying the genetics of adaptation in my lab. We start with QTL mapping just to identify which regions of the genome contribute to a trait difference between strains from our two populations. And then we can follow that up with population genomics, looking at genetic variation, comparing genetic variation between the two populations within the QTL regions that we identify for that trait, and asking which of the genes in that region show evidence for, elevated allele frequency differences between those populations, as you might predict to result from natural selection. And then having identified strong candidates, we can turn to more functional approaches, either in our lab or with collaborators, to try to make more specific inferences about genes or even specific variants that underlie these changes. Today, though, as I've been hinting at, I am really focusing on the genetic architecture questions. And so I'm going to focus in this talk on what we've learned from our QTL mapping studies in particular for multiple adaptive traits. And the way that we do QTL mapping in my lab is using an approach called bulk segregant analysis. And so this is kind of a cartoon of how we do BSA in my lab. We cross a strain from, let's say, a high altitude population and a strain from a low altitude population. We let their offspring interbreed together at fairly large population size for multiple generations, even 20 or more generations, so that there's been a ton of recombination between those two genetic backgrounds, which is really the key for QTL mapping. And then only at the very end of the experiment, in the last generation, we sort based on some phenotype. And then we do pooled sequencing on the individuals with the highest trait value and pooled sequencing on the individuals with the lowest trait values. So that's what we're doing. And I came up with an inference package called SibSam to make better use of this data and how we analyze it, but I'm not going to talk about SibSam today. Instead, I'm going to focus on the empirical results that we've been getting. And so our first study, which was several years ago now, was on pigmentation. The species has evolved melanism on several occasions, including in the Ethiopian highland population that I've been mentioning. And so one of the interesting results from this paper was that, if we took one pair of strains, let's say from Ethiopian highlands and from the ancestral range in Zambia, so the highland population is dark, the lowland population is light. And so, if we do QTL mapping with that pair of that pair of strains crossed, we'll get something like this. But if we pick just a different strain from that same town in high altitude Ethiopia and a different strain from that same town in Zambia, we're going to get largely different QTLs. Now here, there does happen to be one shared in common, which does happen to overlap a known pigmentation gene called yellow. But otherwise, it's a lot more unpredictable, even though there are other narrow QTLs that overlap well-known pigmentation genes like tan and ebony. By and large, you get different QTLs if you do the same experiment, just with a different pair of strains. And this clearly exceeds the differences that we would expect by chance. A lot of these QTLs are strong enough that if they were in the other cross, we should really be able to find them. So, this was a clear hint that at least for this trait, the genetic basis of melanism in the Ethiopian highlands was not due to fixed differences between the Ethiopian and Zambian populations. Rather, there was a persistently variable genetic basis of this trait. And so, it's not just QTL mapping that gives us that idea. If we look at what's going on at the molecular level, so these are in situ hybridizations, staining, protein abundance of these same three pigmentation gene products that I just mentioned, you could see that if you compare this Zambia strain, which is kind of a typical pigmentation, I hesitate to say wild type because they're all wild, but you know, if you compare the Zambia one to either of these two Ethiopian strains, you could see that the Ethiopian strains differ from the Zambia strain in fundamentally different ways. So this Ethiopian strain is most notable in terms of its downregulated expression of ebony, which is a suppressor of dark pigmentation. Whereas this other Ethiopian strain is much more notable in terms of its increased expression of tannin yellow, which are gene products that increase dark pigmentation. So this is not all just QTL mapping results. It's you know, backed up by what we see at the molecular level too. And it's not only pigmentation either. So I'm going to briefly show you a couple of results from projects that were also by Quentin Sprengelmeyer in my lab, showing the same sorts of experiments just with other traits. And that the reason we're doing this on other traits is because we want to have kind of generalizable conclusions about how adaptive evolution is working in this species. So in one case, Quentin did a study on the evolution of elevated ethanol resistance, which exists in European populations and also in some high altitude African populations. But here I'm showing you the same sort of design where, you know, this is a cross between one France strain and one Zambia strain. And these arrows up here indicate the significant QTLs that were inferred by SIBSAM for ethanol resistance between that pair of strains. But if we just take a different France strain and a different Zambia strain from the same local collections originally, then we do the same experiment, then we get basically a almost completely different set of QTLs. So the same thing happens with ethanol resistance. And so this trait too apparently has a variable basis. Same for size. So this is mapping the genetic basis of wing size in Ethiopia. These highland Ethiopian flies are unusually large with especially large wings. And so here, I'm actually showing you 4 different mapping crosses between a distinct Ethiopian and Zambian strains. And again, the pattern is that we see almost entirely distinct QTLs from one cross to the next. And so what we're seeing over and over again is this pattern of a genetically variable basis of adaptive trait evolution, which may reflect an abundance of genetic variation to select upon, and a scenario where not all of that variation needs to be fixed in order to get a population to its new optimum or threshold value. So, you know, the idea is that, you know, this is kind of what variation looks like before selection, and selection is going to raise the frequency of these variants to different degrees. But they don't necessarily need to all get fixed. You don't need all of them in order to have the phenotype that's more beneficial in this novel environment. And so I've been trying to figure out what to call this because I feel like polygenic adaptation doesn't really do it justice. You know, the central feature here is not necessarily that there's a lot of genes involved. Yeah, there's more than a few, sure. But the central feature to me is how it remains variable. So today I'm calling it variogenic adaptation. I'll let you know what I decide to call it tomorrow, or you can let me know what a better idea is during the question and answer session here. So to summarize what I've told you in part 1 here, we've learned that Drosophila melanogaster does still survive in the wilderness of Africa, and that marula is an especially favored food resource. We have estimated the timescale of the species expansion within and beyond Africa. And we've learned about the genetic architecture of adaptive trait evolution by comparing these populations. We have learned that these genetic architectures of adaptive traits are always variable for the cases we've looked at. And that yes, this is polygenic adaptation, but it's not just like a bunch of tiny effects. You know, at least on an individual cross level, there are strong effects present. So we're getting very consistent results in terms of the genetic architecture of adaptive trait evolution. All right, so I'm going to continue in a fairly similar vein in part 2 here, but part 2 is all going to revolve around these same, you know, 6 populations basically. These 3 pairs of warm and cold adapted populations that I'm showing here. So I'm including Zambia as kind of a seventh population here from the ancestral range. And I'm showing, three basically pairs of populations where they're close together geographically, but one is in still a warm environment, more like the ancestral range, and the other is in a colder environment, more like the, more of a novel derived environment. And so we have three pairs, basically, a Southern African pair, an Ethiopian pair, and what I call a Mediterranean pair, which is a little bit further apart, but is actually still pretty genetically similar, as indicated on this genetic distance tree down at the bottom. And so I'm going to tell you about first a population genomic study of these seven really populations. And after that, I'm going to tell you about a couple of RNA sequencing studies that use these same populations to learn about parallel evolution. And so in the population genomic case, what we did is we used a statistic that I had previously developed, kind of elaborating on a prior approach that I was also involved in, that uses three population samples and the allele frequency differences among them. And so the idea is that we're basically kind of polarizing the allele frequency changes that happen in one population's terminal branch here. And so in the case of this study, we're asking, what allele frequency change happened on the branch leading to the cold adapted population specifically, as opposed to its warm adapted partner or the Zambia population. And so we're trying to understand, which genes have a lot of allele frequency change leading to the cold adapted populations, and to what extent is that parallel? To what extent do the different cold adapted populations show elevated allele frequency change at the same genes? So I'm not going to present a lot of detailed results from this study, but this is kind of the best case scenario that we're looking for here. So here I'm showing you allele frequencies at a specific SNP in a synaptic gene called ProSAP. And so what you could see here is in orange, these are the allele frequencies of this particular SNP in each of the warm adapted populations. And in blue, these are the frequencies in their three kind of partner cold adapted populations. And so, the unique result here is that in each cold adapted population, the frequency of this variant is dramatically elevated compared to its warm adapted partner, to an extent that is actually unexpected by chance at any SNP in the whole genome. Now that's just one SNP, but a more general finding from this study is that we actually kind of, in terms of parallel evolution signals like this, we could see that more clearly when we focused in at really SNP level genetic variation. Rather than looking for, parallel elevations in allele frequency differentiation on the scale of whole, almost gene scale windows. In other words, it seems like the width of these elevations of genetic differentiation is very narrow compared with what we might have expected under some simpler models of positive selection. And so we had seen things like this before, even in that pigmentation mapping study that I told you about before. I mentioned that one of the QTLs was over a known pigmentation gene called ebony. Well, this is the pattern of genetic differentiation that we observe at ebony. FST here is basically quantifying how different are the allele frequencies between the Ethiopian highland population and the Zambia population. And what we saw was just this very narrow cluster of very highly differentiated SNPs at this locus. And we did some simulations to show that this pattern was most agreeable with a scenario of a soft sweep from standing genetic variation. So an idea that selection was acting on a variant that was already there and had recombined onto more than one haplotype before selection ever favored it. So this soft sweep idea contrasts with the simplest, most traditional model of positive selection, which I'm calling the hard sweep model here. So in the hard sweep model, you could have selection favoring a newly occurring mutation that is only occurring on one haplotype background. It only has one set of link variants because it just happened. And then when selection increases the frequency of this beneficial variant, the linked variants hitchhike upward in frequency as well. And so you end up strongly altering genetic variation across, a whole region of the chromosome. This contrasts with the expectations from a soft sweep, where you're selecting on genetic diversity that was already around for a while and had time to recombine onto multiple haplotype backgrounds. In this case, if you're favoring this variant, well, it's linked to more than one haplotype by now. And so although you will affect genetic diversity to a degree, the effects are not expected to be as strong. There's still multiple haplotypes surviving here. And so, this is a way that you might get the sort of pattern that we observed here, where, the main difference between a population that's had selection and a population that hasn't is really right at and immediately around the target of selection itself. And so, this motivated us to continue looking for these SNP level patterns of genetic differentiation between populations. Question, yeah.
Be a hard sweep from one of those mutations. How do you know that it's the red circle instead of the ones around it that are like the mutated beneficial mutations?
That's a great question. We don't. I sure wish they looked like this in real data. That would make things a lot easier. But what we really observe is just kind of the pattern, you could even take these ones away. They're not variable anymore. But the rest of the variation too looks different between here and here, but not as extremely. This is still variable if you're looking across populations, I guess. But, I guess even if you just consider this to look like an anonymous variant, that's more like what we have to work with in real data. But the patterns still are what they are. And in this case, there is one particular site which does still show strong allele frequency differentiation. And so, we wanted to keep looking for these SNP level patterns, but we didn't really have a lot of formal justification in the literature for doing this. By far, the convention in the literature was instead to look for whole windows of elevated genetic differentiation. And so Tiago de Silva Ribeiro in my lab did a study to really show that there are cases where you need to do what we're doing here. And so he did a simulation study comparing the power of a window FST against a version where we're just looking at the most differentiated SNP within a window. And he showed that although window FST is better for hard sweeps, either one can detect complete hard sweeps. Those are just easy. So this is showing like what frequency a sweep started at, and this is showing what frequency it ended at. So these are extreme events going from a frequency of 1/2n, a new mutation, all the way up to 100% frequency. Anybody can find that. The window approach can also find, cases where you went from a new mutation up to 50% frequency, let's say. The SNP approach, not so much. But what the SNP approach can do is it can detect complete or mostly complete sweeps where you started from a relatively higher initial frequency. So these are the soft sweeps that I'm talking about. So we've shown that, for soft sweeps in local adaptation, it doesn't necessarily work that well to use window FST, and it can make sense to really pay attention to the SNP level pattern. Curiously, actually, we found that window FST was actually somewhat more enriched in real data in our fly data than SNP FST, which could imply that there's actually a little bit more of these than there are of these going on in our populations genome-wide. And I think that probably has more to do with the partial part here than it does to do with the hard part. I think, our adaptive trait mapping studies too suggested that partial sweeps may be more the rule than the exception in terms of adaptation among these populations. So that really dovetails with that result. All right, so in the next part of this talk, I'm going to shift gears a little bit from all population genetics all the time. And instead, I'm going to talk about some RNA sequencing as well, except I'm going to analyze it again in kind of a population genetic framework. And so here we have RNA-seq data from these same 6 populations. This is data collected and especially analyzed by my former postdoc, Dr. Yuheng Huang. And we have data, to give us both variation within populations in expression and splicing, and also variation between populations, especially between the members of each pair of these populations. And this is only whole organism RNA-seq data, which is kind of a limitation, but we do have it from 3 different developmental stages. In the first part here, I'm going to focus just on data from the colder temperature, because if we're interested in adaptations to these colder environments, well, it would have to show up in that environment to be a successful adaptation there. So that's why we looked there first. And so what we did is we got all the expression and splicing values in terms of intron usage for each of these transcriptomic traits. I'm going to focus basically on expression though in this talk. And so for each of these expression traits, we calculated something called PST, which is basically a quantitative trait analog to FST. So instead of genetic differentiation, we're looking at trait differentiation between our warm and cold adapted populations. So PST will be higher when the expression values from the cold population are all pretty different than the expression values in the warm adapted population. And we identified outliers on the basis of this PST statistic. And so we took the 5% tail there, which is pretty generous, but we're not necessarily counting on every one of those being a true positive target of adaptive regulatory evolution. What we're going for here is just to have a decently sized group of outliers that are strongly enriched for targets of real natural selection. And so on average, we found that there was a clear signal of parallel transcriptomic evolution here, in that there were more than twice as many shared PST outliers between population pairs than we would have expected by chance. Although I do note that the level of sharing differed quite a bit depending on which 2 pairs we're talking about and which developmental stage we're talking about. We found much more consistency for adult and to some extent larva, and much less parallelism at the pupal stage, which actually aligns with some other published evidence about how important each stage is to cold adaptation. To give one kind of notable example, there was one gene that was at least a moderate outlier in all 3 population pairs, this IML1 gene, which has a few intriguing roles. Like a lot of genes, it does more than one thing though, but this could be an interesting target for future studies. Another question we were interested in this study was with regard to whether adaptive regulatory evolution proceeded more by cis regulatory changes or trans regulatory changes. So if you're not familiar with that dichotomy, what cis regulatory change means is that it is governed by genetic changes at that locus, like a SNP in a promoter or an enhancer. Whereas transregulatory changes are encoded by a factor that is somewhere else entirely. So like a transcription factor, maybe gene A is differentially expressed between these 2 populations, but it's not because of any change at gene A. It's because they have a genetic difference at gene B, which is a transcription factor, and regulates those genes differently because of its change. So those are the changes. Those are the 2 scenarios that we're interested in testing in terms of which one contributes more to adaptive regulatory evolution in particular. And so, the way that we can study that is to do a cross between our 2 strains that have a different expression level. And if the cause is a cis regulatory variant, then we expect to see a difference in allele specific expression between the alleles from the 2 populations. The one with this promoter variant gets expressed more than the one with that promoter variant. Whereas if the cause of the parental strain difference was a trans factor, then the 2 alleles should actually be expressed the same in the F1. There's the transcription factors are what's mediating the parental strain difference, and they can't tell the difference between the two parental alleles because there is no causative change there. So by an analysis of allele-specific expression in F1s compared to parental expression, we can make inferences about cis versus trans expression. And we developed a new analysis method to be able to do that on strains that are not 100% inbred across the whole genome. And so in terms of the results of that analysis, what we found was that, well, it depends on the population you look at. Here, I don't have a consistent answer for you. So for example, the Mediterranean pair seem to proceed more by adaptive cis regulatory evolution, whereas the Ethiopian pair seem to proceed more by means of trans regulatory evolution. And there were some other interesting correlates of that in the Ethiopian sample. And one of them is that the outliers, the PST outliers from the Ethiopian sample tended to be co-regulated with more other outliers. So, there's hints that there's been some important shifts, maybe at nodes in the gene regulatory network that have created, more than one outlier apiece even perhaps. And so, you could see this elevated co-regulation signal in the Ethiopian pair specifically. So this is co-regulation based on the variation within the cold population specifically. One thing that was a little bit more consistent though is that, between the cis outliers and the trans outliers, and by outliers, I mean candidates for adaptive regulatory evolution, the ones that were encoded by trans factors were more likely to be shared between different population pairs. And so, I don't know if that reflects, more standing variation for trans regulatory variation or some other reason, but that was the observation. And a final observation here is that for the cis outlier genes specifically, we saw on average that they had higher genetic differentiation, which you would actually expect. So the idea behind a cis outlier is that adaptive evolution acted at that gene, not at some other transcription factor. And so we should have had allele frequency change there. And on average, we did in all three population samples, although they're not all significant. And it depends whether you're looking at window FST or SNP FST. The overall signal, though, is pretty consistent for this kind of elevation. So this is just the proportion of either cis outlier genes or non-outlier genes that might be evolving just neutrally that are within kind of the tail of our FST distribution. Okay, I'm going to tell you about one other RNA sequencing study here, and that has more to do with phenotypic plasticity. And so we're interested in asking whether gene regulatory evolution, in the case of potential adaptive changes, was following along the same lines as phenotypic plasticity, or else actually opposing it. And so what we did here is to add data from a warm environment to the cold data we already had. And then we were asking, in cases, so if you think about what we call like the naive plasticity of the warm adapted population, so what happens when you just take that warm adapted population and throw it into a cold environment that it's not used to? What happens to expression? Well, that's plasticity. And then we can ask, well, what happened on the evolutionary side in the cold adapted population? Did it accentuate that initial plasticity, or did it actually neutralize or kind of overkill, reverse it? And the very consistent pattern we found in that regard is that adaptive evolution tended to oppose the initial phenotypic plasticity. This was true, to some degree in all three populations for both expression and splicing, although just 2 out of 3 pairs were significant in each case. And so there are a couple of possible explanations for this. To some extent, adaptive evolution may be correcting plasticity, which was maladaptive in the first place. Maybe some of those plastic responses are just not a good idea, and you want to get expression back to the universal optimum. On the other hand, some of this could actually reflect, the idea that some of what we're calling plasticity may actually reflect sort of a stress response in the warm adapted population. Whereas in the cold adapted population, their adaptation may be sufficient to avert that stress response. And so you see the cold adapted population kind of back, near where the expression started out. That would be, an adequate explanation as well. So I think for the sake of time, I'm going to skip the decanalization part of this study, but you can ask me about it later if you want. To summarize what I've told you about in this part of the talk, we talked about the idea of soft sweeps and local adaptation requiring a SNP level approach to genetic differentiation. And we looked at both population genomic and transcriptomic data to look at evidence of parallel evolution between cold adapted fly populations. We found that either cis or transregulatory variation could play a leading role in adaptation depending on the population, and that adaptive evolution tends to neutralize the initial plastic changes in expression. So I want to save at least a few minutes here to tell you about the last little part of my talk, the time travel part. This began when I came to UW, and I was kindly given a set of fly strains, which were more than 60 fly strains that were all collected from the same time and place. They happen to have been from Providence, Rhode Island, and to have been collected within about a 9-year time span, initially collected by Margaret Kidwell and maintained very effectively over the years by Rayla Temin here. And so I want to give credit to both of these illustrious geneticists for making sure that these flies were collected and maintained through time. And so what I wanted to do with these flies was to compare their genomic variation to what we would see today in the same location and ask how much has changed over this roughly 30-year time period. 30 years may not sound that long, but we're talking about roughly 500 generations of fly evolution. And so that's what we did. We compared these 60-some old strains to pool-seq data from hundreds of new flies, both from fall and from spring, trying to kind of control for seasonal evolution, which is known to exist. We did a bunch of quality control, especially for the old genomes, to make sure that they looked like they were what we thought they were. 2 of them actually looked like clones of the same line, and so we discarded one, but the rest looked pretty good. And so then Jeremy Lange in my lab led an analysis of what changed over these 500 or so generations. One thing he did initially was to try to estimate a population size. The estimate he got was actually surprisingly low for a species with kind of a global NE that's thought to be in the millions. The local NE estimate that he obtained was less than 10,000. And this could be an underestimate for a couple of different reasons. This is assuming that all of the allele frequency change was due to genetic drift and not to migration or natural selection, for example. But it provides, even if it's an underestimate, it gives us kind of a conservative null hypothesis in terms of the allele frequency changes that we might expect at any given place in the genome over these 500 or so generations. So, when we looked at allele frequency changes through time, we can ask what we would have expected based on this number. And actually, there were a couple of loci in the genome that were more extreme than anything we would have expected by chance. And so what we did was a very similar approach to what I've talked about before, using this population branch approach to zoom in on one branch in this 3 population tree. So here we're using our new spring and fall samples to kind of wash out the seasonal effects and focus on the long-term effects between old and new samples. And so this is a genome-wide plot of this population branch statistic across the whole genome. And I mentioned there were a couple of genome-wide significant loci. One of them, of course, is this highest peak here, which is a pair of tandemly duplicated cytochrome P450 genes. In fact, these genes are known to have a role in insecticide resistance, and there is a known copy number variant at these genes that has been correlated with resistance to a specific insecticide called deltamethrin, which I will mention was discovered in 1974 and commercialized shortly after. And that timing is quite relevant because what we observed, to our great surprise, was that there was actually a considerable leap in the frequency of the resistant associated variant with both genes present, even within the sampling years of the old strains. So even between 1975 and 1983, it's jumping up from near zero frequency potentially to almost 50%. And curiously, though, it never did, or at least by the present, it didn't really get any higher than that, which might reflect the trade-offs that insecticide-resistant alleles often have. But even just based on this rapid rise within the old samples, we were able to use an approach based on approximate Bayesian computation. This is a simulation-based approach to estimate how strong was the selection that governed the rise of this allele over this time scale, and what do we think, what sort of frequency do we think it was initially starting at? And this is actually a pretty strong selection coefficient compared with what we tend to encounter in real life. And the analysis does suggest that the variant was already there. And so that's not surprising, and including based on the whole, soft sweep evidence that I've offered earlier in the talk. So this is at least one example of the sort of thing we could do on a locus specific level. We were also interested in more genome-wide perspectives on, to what extent do we have an enrichment of high genetic differentiation? And so we did what I would call an outlier removal approach. So, lumping together nearby outlier windows that probably reflect the same instance of natural selection, we asked how many of these regions would we need to take away to erase the genome-wide enrichment that we observe either on the whole window level or on the single SNP level for genetic differentiation. And the answer for window PBS is that actually we only need to remove 4 regions to get rid of that enrichment. Not wildly impressive, but interestingly at the SNP level, we'd have to remove fully 20 regions across the genome to get rid of the enrichment of SNP level population branch statistic excess. And so this could imply that there are at least about a couple dozen loci with strong enough selection over this 500 generation time span that we can infer it with this sort of approach. And the fact that it was stronger evidence on the SNP level could again suggest that a lot of this selection may be on standing variation as opposed to new mutations. So what we're doing next, which I'm not planning to fully tell you about today because I've gone a little bit long here, is to actually extend to the time span of a few thousand generations. And so our collaborator, again, Marcus Stensmeyer from Sweden, managed to get DNA out of 24 flies that had been sitting in a museum drawer for 100 or even 200 or more years. And so we're comparing their genomes to those from modern flies from the same location. And this analysis is ongoing. So I'd be glad to give you a preview of those results sometime, but for now, we're running a little short. Obviously these temporal samples give us some unique opportunities in positive selection to study natural selection on both the single gene and genome-wide scales. So with that, I would just like to thank the people who really made this work possible, both members of my lab, past and present, and collaborators, and also the funding agencies and the great environment that I have here at UW-Madison. So thanks a lot. Sorry for running a bit long, although we got a little bit of a late start too. And I would be glad to take any questions, but I totally understand if anyone has to run. Yes, Carol.
Hey, John. I really like the talk. What a nice comprehensive story. I have a couple of questions. One's regarding your conclusion that the affirmatory shift, the plastic shift, the adaptive shift in mitigating might be due to either stress or
plasticity was originally maladaptive in some cases.
But there's an alternative. If you look at physiological literature, there's an alternative explanation. This pattern arises quite often when there are compensatory effects, when there are other adaptive compensatory effects that don't require that phenotype to not be as great. So I can give you several examples, like from osmoregulation or
So you're suggesting that maybe the initial plasticity was adaptive, but after genetic adaptation, it was no longer needed?
Yes.
Ah.
That happens quite a lot.
Uh-huh.
For instance, if you look at evolutionary shifts in human ophthalmology,
Mm-hmm.
it often has to go up initially in the early stages, but then later over time, because of greater technique, protection, it goes down again. So you see that
Yeah.
a lot in plastic evolution, where you see that quite a lot in physiological adaptation cases where the initial plastic shift has to be really strong.
Right.
Then over
Mm-hmm.
time, there's compensatory adaptations
Yeah.
that allow it to not be as extreme.
I wonder if the stress response idea that I gave is maybe more of a particular case, and you're giving more of a nice broader model for that. Yeah.
In general.
Mm-hmm.
There is the heat shock response and the chaperone response, but I'm not talking about that.
Mm-hmm.
I'm talking about the evolution of actual traits.
Yeah.
And I can give you examples.
Sure.
But yeah.
Definitely. I would be glad to hear more about that.
Or why you see that pattern.
Thank you. Yeah.
My second question, your result with the cis-regulatory evolution is quite intriguing, where there's a greater FST for the genes under cis-regulatory evolution. Is that what you found?
Yes. We did find elevated FST at the cis outliers. Mhm.
And those are non-synonymous changes?
No, not necessarily. We're looking more agnostically in terms of site functions, and we're just asking, for each cis outlier and each non-outlier, is that locus an outlier for window FST, and is it an outlier for maximum SNP FST, where the maximum SNP could be any type of site?
Oh, is it enriched for non-synonymous? I'm just curious.
I don't actually think we looked at that question exactly. It just had to be in a defined target window, within or around the gene itself. But that would be interesting to take a closer look at. Mm-hmm. Right. Yes. It would be fascinating to try to figure that out. It would probably take some work to try to disentangle, whether one type of change was just hitchhiking with the other. But if we could separate those possibilities, it would be really interesting. Thanks. Yes, Max. Mhm.
So I know you, because of time constraints, you lost over the section on analyzation, but you had a concluding slide which stated that natural selection reduces anti-plasticity, which we did talk about. But then in the same sentence, he also said that it tends to decrease chemicalization.
Yes.
That statement seems to contradict on some of your aren't you increasing chemicalization if you're decreasing the plasticity?
Not necessarily. We're actually defining canalization more in terms of genetic robustness, not environmental robustness in this case. And so we're interested in a particular hypothesis that we had generated in a previous study based on the Ethiopian of larger wing, the evolution of larger wings in Ethiopian high altitude flies. So what we had noticed is that the high altitude flies had not only larger wings, but also more wing vein abnormalities. And we wondered, the hypothesis that we came up with is that, this trait, like many others, might have been buffered or canalized to some degree in the ancestral range. But when evolution went and changed that trait, to some degree, it may have undermined the buffering mechanisms. Not all of these buffering mechanisms might successfully buffer the evolved trait. And so we wanted to ask whether that might be true for adaptive gene regulatory evolution as well. And indeed, we did find evidence for that for our outliers, for expression anyway, the targets, the potential targets of adaptive expression evolution were easier to destabilize, in this case, via inbreeding. So the outliers were more destabilized by inbreeding than non-outlier genes.
Okay, so it's independent of robustness and chemicalization of the environment.
Right. We specifically looked at genetic robustness in this study. But we found that indeed adaptive evolution did seem to undermine that. I know we had a couple of questions from students further back. Mhm.
Yeah, I have a question. When you collected wild flies from wild environment, is there any way to make sure that they represent the whole population, the local population? And then when you get those strings and lab, do you use backbones to make them wild instead of a inbred?
Yeah. And I mean, for some of our specific experiments, we do specifically outcross things. Like for a lot of the RNA-seq data, we specifically did that. In terms of the wilderness flies that you're talking about, we actually don't have any wilderness fly lines in the lab yet. We had just been sent some preserved flies for that study. But in a sense, I'm not sure they do reflect the whole population. We found surprisingly that the wilderness population sample had slightly lower genetic diversity than the town populations, which doesn't really make sense if town populations were founded from wilderness populations. So our suggestion is that in the wilderness, flies may exist in more of a metapopulation where you have small local demes. And each local deme doesn't necessarily contain all of the genetic diversity that you would see in the broader population. But that is a hypothesis that remains to be tested based on further sampling of wilderness flies.
Why does it matter if the adaptation was cis or trans regulatory?
Why does it matter? This is really just a basic evolutionary question, like most of the questions that my lab investigates. We're interested in, what types of genetic changes in general tend to fuel adaptive evolution, and whether, there's the idea that probably gene regulatory evolution is very important in adaptation overall. Like based on population genetic evidence, it looks like probably most of the adaptive fixations of sites across the genome are at non-coding sites, like a vast majority. So that's probably a lot of the adaptation is going on, probably relates to gene regulation in one way or another. But what kind of gene regulatory changes really fuel that? And although there had been other studies on cis versus trans evolution, ours was the first to relate it to specific evidence for adaptive evolution as opposed to just neutral evolution.
Would it be fair to say that trans evolution tends to have a larger impact than cis evolution?
Yes, it's true. We didn't really talk about the magnitudes of the changes here, and we even hinted that maybe multiple trans outliers might even reflect the same genetic change in terms of like the co-regulation among outliers, especially in that Ethiopian sample that had more trans contributions. So it is entirely possible that one type, potentially trans, may be more consequential on a per genetic change basis than the other.
Let's thank John one more time for sharing.
Thank you all.